Okay, good people. So, welcome to the second part of genetics problem 2021. This is just national examination genetics problems 2021. So I had that solving in the first part. So today I'm going to concentrate with the second part, and mainly the second part is concerned with those crossing. In the first part I concentrated with how to generate the symbols. So today I'm going specifically on a crossing to show exactly what is happening. So in my last presentation I was already talked about this cross here. I had the symbols as usual, and then I presented the first cross, which was a cross between, now in the first cross was a cross between a black haired male and white haired male, and they were from pure line. So I had this one versus this one. So now what is important is the question wanted us to use Punnett square. So if it wanted to use Punnett square, so we cannot do that simple cross. I did this because actually I was out of time, by the way. So I was giving a frame of what is going to happen. So basically we're supposed to do something like this. In order to complete this cross, we already have those gametes, so we have to do the independent assortment in order to form, we had the genotypes, so we had to do the independent assortment, which is meiosis during formation of gametes, whereby anyone may combine with either one of another pair. Then we will have this BG in a gamete cell, then this one with the second one. Now we'll have BG in a gamete cell. Now the same thing with this, we'll have BG in a gamete cell. Then this with the last one, we'll have BG in a gamete cell. Now the same thing with the second. Now in the independent assortment on the other parent. Now this one, then we'll have this with the second one. We'll have BG then in a gamete cell. Then the second one with the first one, we'll have small b, small g in a gamete cell. Then the last one, as usual, we'll have small b, small g in a gamete cell. I think you know what's going on there. So after having those gametes, we must draw the Punnett square. So maybe let me have some space there. So let me clean and have some space for drawing the Punnett square. I have to draw the Punnett square, So now what I'm going to do here is having the Punnett square. So the Punnett square will be something like, now the Punnett square I think everybody knows. So here will be the Punnett square. Now the Punnett square so has got 1, 2, 3, 4. Now we have 1, 2, 3, finally 4. So now this is Punnett square. And now in my Punnett square, so I will put those gametes. So assume one parent here was male and the other one was female. So I think, I don't know if it was presented there, was conducted in pure breeding variety of goats, the black haired male, you see. So we had the black haired male. So in my Punnett square there, I present sex. Now on the upper side I will have gametes for male parent, and on the other side I will have gametes for female parent. So after having that, I'll put those gametes. Now male has got caps B, G, gamete, B, G, gamete, B, G, gamete, then BG, gamete. Now these are the gametes. Now female has got BG, now they are small gamete, BG, then gamete as usual, BG, gamete, then BG, gamete. Now these are the gametes. So next is fertilization, because question wanted us to use Punnett square, but if it wasn't that recommendation of using the Punnett square, we will have that simple way of using genetic crosses. So now in our Punnett square, now everything will be B, then small b, G, then small g. So B, small b, G, small g. Everything is there in that way. So this will be our Punnett square. Now everything will be in this way. Now the whole Punnett square will be covered with those similar types of genotypes. Now similar types of genotypes will cover the Punnett square. So it's very simple, like this. Now the Punnett square is covered with everything in the same way, like this. Now the Punnett square. Now the Punnett square like this. The Punnett square like this. Now the Punnett square like this. the Punnett square, and the last one in the same way. So now these will be the genotypes now. In the Punnett square, these will be the genotypes. So now what will be the phenotypes now? If we have genotypes, what will be the phenotype? Phenotype, now the phenotype, and this will be F1 phenotype, those individuals of the first generation. So you'll find that this genotype has got this caps B, and then has got this G. So B was for black haired, and G was for gray haired. And as long as we had the idea that B is epistatic to this G, so when there's caps B and caps G, now in the phenotype only caps B will be expressed. So all individuals, all the F1 were black. Now they were black haired. Now all the F1, they were black haired. So this is, now it is black, you see. Okay, let me write in a correct way. So now all the F1 will be black haired. So it's something like this, black haired, all the F1. So now the idea was in a question, however you find that now in our question there was F1 male versus white haired female goats were crossed, and the F1 were black. So all black F1 individuals, they had this type of genotypes. Now all the F1, they had these genotypes. Then now things goes on whereby F1 were saved to produce F2. Now let us see a cross to produce F2. So now a cross to produce F2, it means we will start with those individuals there, those genotypes there. So all the all the individuals involved in the second cross will be black haired. So our second cross will be something like, our second cross will be parental phenotype. It will be a cross between black haired male versus black haired. Now it will be a cross between black haired male versus black haired female. A black haired male versus black haired female. So now in their genotype, parental genotype, they will have similar genotypes. Now caps B, small b, caps G, small g, they are black. Caps B, small b, caps G, small g, they are black. Then next is meiosis. Now meiosis is all about gamete formation. So during gamete formation, now we have the independent assortment. Now this is gamete. Now this is gamete as usual. Now this is gamete. Now with the last one is gamete now. So they have similar genotypes, so similar gametes will be formed. Now we'll have the same thing on the other side. Now this one, the same thing now, things goes on. So we'll have B, then caps G in any way. Now this one, yes. Then we'll have this with the last one is small b, and then small g. So now that's how things goes with those we call the gametes. So you label there as gametes. I think you get the idea. You label the gametes. Now next is fertilization. So fertilization as usual, we have to use Punnett square. So during fertilization, it's where Punnett square comes in. So now maybe I can clear this. I need to have a Punnett square at the same place. I'm going to have Punnett square at the same place. Punnett square at the same place. Punnett square at the same place. So now this is just the Punnett square for F2 individuals now. Punnett square for F2 individuals. So now things will be like, let me have the Punnett square first. The Punnett square like this: 1, 2, 3, 4, 1, 2, 3, finally 4. So we have those things there. Now after having the Punnett squares usual, we will present those sex. Here is male, female on the other side, so we put gametes there. So now male gametes, now we have the first one, second one, and until the fourth, the same thing there. So now caps B, caps G, gamete, caps B, small g, gamete, small b, caps G, gamete, small b, small g, gamete. caps B, caps G, gamete, caps B, small g, gamete, small b, caps G, gamete, small b, small g, gamete. So you see, so we have got the gametes. Now next is fertilization. We'll have B, B, G, G. So you must be careful about those letters. You must present as how they appear if it is caps or small. So make sure that you consider that. And then we'll have B, small b, then G, small g. Then the same thing, G, small g. The same thing goes on, small g. The same thing, small g. Now things goes on and everything is small. Then we carry on. Yes, we have everything is caps there for G. Then we'll have B, small b, caps G, small g. Then next there is small b, caps G, I think you understand. So B, B, then G, small g. Then you do the same thing there. B, B, then G, small g. Then we have caps B, small g, small g. Small b, caps G, small g. The last one now all the time is just everything is small. So now we have to interpret those genotypes in a Punnett square to see what are the possible phenotypes from those genotypes in a Punnett square. So basically we will obtain the ratio. We can obtain the ratios now from those genotypes. As I said on the guidelines of ratios, we must identify the genotypes and then ask ourselves how many genotypes produce the same phenotype. So that's what we're going to do there. So now we have the first one. So remember, we had this caps B for black, we had small b for white, we have caps G for gray, small g for white. So in order to have black, we must have at least a single caps B. We will have black. So let us see, at any place in those genotypes where there is caps B will be black. So how many black are there? So let us count how many black are there. So now we can count with this. Maybe black 1, or maybe let's do that common way now. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now we have got 12 now. Remember, we have 12 for black. Now we'll put there 12. And then let us see the rest. Now let us see the rest. Now the next thing is, let us see what is another genotype. Maybe this one. You see we have small b and then we have caps G there. So now when we have those b's in a recessive form, then G is caps. Now it is a time when this G is expressing, now it's a soft. So this G is for gray. So let us see how many gray will be there. Let us see how many gray are there. So now gray actually must have at least a single caps G. 1, 2. We have only 2. Now this will be the third. There are only 3. Now we have only 3 are gray. We have 3 for gray. Now the remaining is this one is recessive for everything. So if it is recessive for everything, we had this small b for white, then G for white again, or sometimes maybe one of these may have no effect on the phenotype. So now the last one will be just a phenotype for white. Now we will have only 1 for white. So we have just obtained our ratio. That was 12 ratio 3 ratio 1, which is just a ratio for simple dominant epistasis. So that's how you're supposed to answer this question. And you are not supposed to cross like this. Don't do what I'm doing here, this one, crossing. Just count them without crossing. Always you do those crossing in order to avoid repeating when we are counting those genotypes. So remember, don't use that idea of crossing those things. It's not correct in your answer. It's not correct to do that. So we have done with this, and in the next video I'm going to solve national examination in 2022, which was just within the same pattern, that was epistasis, but it came in a different design to an extent that someone may not recognize that it is the same thing. So thank you for your time. Make sure that you put a comment if you have any question, and also you can like this video and you can share if you think it's going to be helpful to another person. Thank you.